let us move to the second phase of our conversation today and introduce you to the basic concepts in pharmacokinetics and its clinical applications. We will talk about the apparent volume of distribution and the clearance parameters. These are two parameters that we call primary pharmacokinetic parameters. Then we will address first-order kinetics. The vast majority of drugs that we use in clinical medicine follow the pattern of first-order kinetics of drug elimination, but there are exceptions, and that would lead us to discuss Michaelis-Menten kinetics for drug elimination. So, pharmacokinetics, the quantitative analysis of the time course of drug absorption, drug distribution, drug metabolism, and excretion or elimination from the body. Schematically here, we prescribe a dose or administer a dose of medication to a human subject. Then we need to wait for the process of absorption to take place so that the drug can be carried typically from the gastrointestinal tract to the systemic circulation. The drug in plasma may circulate as the free drug, but also may bind to plasma proteins like albumin. And again, you have this reversible equilibrium between drug that is free in plasma and drug that is protein bound. The extent of protein binding varies tremendously depending on the drug in question. Then drug elimination will take place, but of course drug distribution from the plasma compartment will take place. The drug may actually distribute to most tissues, and you may find non-specific binding of drug to tissues, but where we're really interested is in the distribution of the drug to its site of pharmacological action, what we call the biophase, and of course the study of receptor binding, and ultimately the effect of the drug that we're looking for. Now again, drug metabolism may contribute to elimination, and renal excretion is a pathway for elimination of drug metabolites, but also a significant pathway for elimination of the parent drug itself if the biotransformation is incomplete or actually does not take place. And finally here, we want to measure the element of adherence. Physicians prescribe medications to patients. Ultimately, patients decide whether or not they will take the prescribed medication. Monitoring for adherence is critical in the process of drug development. If you are evaluating the efficacy of the drug, you want to know that patients are actually taking the medication as prescribed before you make a statement like the drug does not work. Well, we need to have rigorous control for adherence in the context of clinical drug development. So what are the uses of pharmacokinetics? Pharmacokinetics provides the basis for rational dose selection in therapeutics. It is essential for the development and evaluation of new drugs. We need to know how drugs are absorbed, to what extent they are absorbed, if given orally, where does the drug distribute, and again, how is the drug eliminated, and what is the rate of drug elimination. Pharmacokinetics is also very important in basic studies of drug distribution in animals and humans with the use of PET scanning, positron emission tomography, where you can actually visualize the binding of drugs to its site of action. Now, a central tenet of pharmacology is the dose-response relationship. We carefully study drug exposure-response relationships in order to find the right dose for a given therapeutic indication. Now, exposure response, of course, applies to both drug efficacy and toxicity. It is important to understand the range of doses that are useful therapeutically and the range of doses and resulting plasma concentrations that may lead to toxicity with the use of this drug. Now, there are a number of pharmacokinetic, pharmacodynamic modeling approaches that have been used to define these drug exposure response relationships, and you will deal with that in subsequent sessions of the course. Now, linked to this notion of the dose-response relationship is the target concentration strategy that has been very useful clinically for a number of drugs. We already addressed the concern with individual variation in drug exposure when drugs are used in standard doses, as we saw with pioglitazone or metformin. So this approach, the target concentration strategy, attempts to individualize therapy when therapeutic and toxic ranges of drug concentrations in plasma have been established. This is important to define a useful therapeutic range and then to target therapy to that range of therapeutic concentrations. The ultimate goal is to optimize efficacy and minimize toxicity. Now, the first description of therapeutic drug monitoring that we have on record is that of Dr. Wuth using bromides and establishing ranges of therapeutic concentrations of bromide as a sedative. Of course, this approach is now used for a number of drugs, and for example, lithium carbonate in bipolar disorder is administered with very strict attention to the resulting plasma levels of lithium so that you maintain efficacy and avoid some potentially serious toxicities with the use of this agent. Now, what drugs are candidates for therapeutic drug monitoring? Generally, drugs with low therapeutic index, meaning that we can quickly move from concentrations that are therapeutic into ranges of concentrations that can cause toxicity. The example of lithium is a very good example of a drug with a low therapeutic index, but there are many others like digoxin and some antibiotics. But in any event, that category of drugs is a good category of agents for therapeutic drug monitoring. You may also be dealing with a clinical situation where you don't have, if you will, physiologic endpoints that you can observe on an ongoing basis or biomarkers to guide the dosage. You may be dealing with patients with a seizure disorder, epilepsy, where the seizures are infrequent and, of course, undesirable. So you use the range of therapeutic concentrations, if you will, as your biomarker to guide dosage and hope that that would lead to a significant reduction in the frequency of seizures. We already stated that pharmacokinetics vary widely between individuals. So if you have a target concentration, then you can adjust doses on an individual basis. Occasionally, we may use a measurement of plasma drug concentration to monitor adherence, but there are some issues with this approach as well. So let's see schematically then what happens when using the target concentration strategy. We have an estimated initial dose that we administer with a target level in mind. Some drugs need a loading dose to establish a therapeutic concentration quickly, followed by maintenance dose. Other drugs we begin simply with a maintenance dose. Therapy is initiated, and then we have to evaluate the patient. We need to see the response in the patient. And we may also measure a drug level, and based on this assessment then we may refine the dose estimate, adjust the dose, and then continued on an iterative basis to optimize the range of concentrations that we want to maintain throughout therapy. Now, how do we choose a target level? Well, this is an empirical process in terms of defining what ranges of concentrations are therapeutic and where you have minimal or no toxicity. So we will have the example of digoxin to address this topic of how do you define a therapeutic range of concentrations. This was a study conducted in Boston by Dr. Smith and Haber in patients that were being treated with digoxin because they had either congestive heart failure or atrial fibrillation requiring rate control. And what they saw, looking at a group of patients that were classified as being toxic or non-toxic based on clinical characteristics and electrocardiographic characteristics without knowledge of the resulting digoxin levels. And this is a histogram of the distribution of concentrations of digoxin in the patients that were non-toxic, and then higher concentrations of digoxin being measured in patients that were clinically toxic. So on the basis of these empirical observations, then a therapeutic target range is proposed. In this instance, 0.8 to 1.6 nanograms per ml of plasma. It was considered that levels in the range of 1.6 to 3 nanograms per ml were possibly toxic, and patients that had levels of 3 nanograms per ml or greater were probably already having digoxin toxicity. But once again, based on further evaluation of the effects of digoxin, not only on function in patients with congestive heart failure, but now in terms of survival after long-term treatment with digoxin. This study that was published in the early 2000s, looking at patients on therapy for congestive heart failure and receiving digoxin throughout this period of observation that lasted 48 months, and then looking at survival on the basis of the observed levels of digoxin. Now, there was a placebo group here that you see with the continuous line, These patients were receiving treatment for congestive heart failure, but were not receiving digoxin as part of their regimen. And then patients that were receiving digoxin, but now stratified based on their digoxin levels. Low levels of 0.5 to 0.8, intermediate levels of 0.9 to 1.1, and high levels greater than 1.2 nanograms per ml. Now, you see that survival changed based on the digoxin levels and the range of digoxin levels that were measured actually at 1 month into the trial. 1 month into the trial. The better survival is actually in patients that have low digoxin levels in plasma, and there is a disadvantage in terms of survival for patients that continue digoxin and maintain a level, or at least had a level 1 month after beginning the trial that exceeded 1.2 nanograms per ml. So, of course, the question is, what were the digoxin levels well into the trial? We don't have that data, but based on this survival analysis for the use of digoxin in patients with congestive heart failure, there is a new therapeutic range that has been proposed, namely 0.5 to 0.9 nanograms per ml, much lower than what was usual in clinical practice. And the benefits may result from inhibition of sympathetic nervous system rather than improved inotropy or improved contractility of the myocardium. There are limitations for the study. We already pointed that out, that no digoxin levels were done after 1 month in the study, and considering that the observations lasted for 48 months. So that's how we estimate a target level. And then in the case of drugs that require a loading dose, and that was the practice actually with digoxin, we need to estimate the loading dose based on the concept of distribution volume. Distribution volume, or apparent volume of distribution, a primary pharmacokinetic parameter. So let us use the example of digoxin once again. Here we're plotting the concentrations of digoxin in plasma. This is a logarithmic scale versus time in a linear scale. And we're showing the plasma concentration versus time curve for digoxin after intravenous administration of 3/4 of a milligram. Single dose. This is a loading dose. And now we see that the plasma concentration versus time, plotted semi-logarithmically, declines in a bi-exponential fashion. We refer to this as the distribution phase, and then this terminal phase we call the elimination phase. Now, the modeling here is plotting the tissue concentrations of digoxin over time, and we see that those tissue concentrations of digoxin rise as the plasma concentrations of digoxin are declining. Now, in order to estimate the apparent volume of distribution for digoxin, one approach is that of the extrapolation method, namely extrapolating from the terminal phase of this curve back to time 0 and estimated this C sub 0, or initial concentration of the drug. Now, that is again one approach to estimating the apparent volume of distribution, and we are using what we call a single compartmental model of drug distribution and elimination. We administered the dose. In our example, we gave this dose intravenously. then we have this single body compartment, a hypothetical compartment where the drug is distributed, and then we are showing here the parameter of elimination clearance. And basically what we're doing in this example, the volume of distribution by extrapolation is estimated as the ratio of the dose over that extrapolated initial concentration. The assumption, of course, is that instantaneous distribution occurs. We saw that that is not the case, but once again, this is one approach that has been useful in terms of estimating the apparent volume of distribution. There are other approaches that you will discuss later in the course, the volume of distribution by area and the volume of distribution at steady state. So, the example of digoxin, initial digitalization, this is a term referring to the loading dose of digoxin, 1/4 milligram being administered, and that distributing into a single compartment, resulting in that initial concentration of 1.4 nanograms per ml. You see here we are doing our proper dimensional analysis in terms of the dose that was administered, the measured concentration in plasma in terms of nanograms per ml, and then applying that principle, the dilution principle, if you will, we have now our dose in nanograms per ml, our concentration in nanograms per ml, and we have this rather large volume, apparent volume of distribution of 536 liters for digoxin. Of course, this does not agree with the reality of physiological body fluid compartments, but nevertheless, the apparent volume of distribution is a critical and very important pharmacokinetic parameter to determine. Now, let's go back to the process of drug distribution. We saw that distribution, in fact, was not instantaneous, and that has an impact on the action of the drug. In this case, the chronotropic action of digoxin, in that digoxin slows the heart rate. Here we're looking at ventricular rate in a group of patients with atrial fibrillation with rapid ventricular response, and we have both oral and intravenous administration. This is from the classic work of Harry Gold and his co-workers in the early 1950s. And what we're seeing here is a significant reduction in heart rate after the intravenous administration of digoxin, but you see that the effect is not instantaneous. The maximal effect, in fact, requires 6 hours before we can observe that significant slowing of the heart rate in patients with atrial fibrillation. So drug distribution may in fact impact the onset of drug action. That is, the rate of drug distribution may impact the onset of drug action. So now, if we want to continue treatment, we have to select the maintenance dose. So what is the principles that applies here? Now, in order to estimate the maintenance dose, we need to understand the concept of elimination half-life and elimination clearance, clearance being the other primary pharmacokinetic parameter we referred to a moment ago. So, simple definition, elimination half-life, the time required for the plasma concentration or the total body stores of the drug to fall to half of the concentration or amount present at some previous time. So a very straightforward definition, but again, half-life applies strictly to drugs that follow first order or exponential kinetics of elimination, and we will come back to this in a moment. So let's look at some simple equations here referring to the half-life, again assuming first order kinetics of elimination, and the half-life can be estimated then as the product of the natural logarithm of 2 times the apparent volume of distribution divided by the clearance of elimination for that drug. The first order elimination rate constant can be estimated as the ratio of the natural logarithm of 2 over the observed half-life. And finally, the elimination clearance can be calculated as the product of K times the apparent volume of distribution. But in fact, K does not determine clearance. This is one way to estimate the clearance of elimination, but in fact clearance determines both the half-life and the first order rate constant. Now, maintenance therapy in the case of digoxin. Now, how much do we need to give in order to maintain that therapeutic level that we were looking for, in this case 1.4 nanograms per ml? Well, we need to estimate how much drug is lost over time. In this case, it was estimated that 1/3 of the total body stores of digoxin is lost daily. In the case of digoxin, the drug is eliminated primarily via the kidneys. So 1/3 of the total body stores at time 0, namely a quarter, or rather 3/4 of a milligram, 1/3 of that is a quarter of a milligram. So that is the daily loss, and that is the loss that has to be replaced on a regular basis. So that's how you establish what your maintenance dose should be. Now, you may start treatment without giving a loading dose, and this is a brute force demonstration of the fact that drug accumulation will take place over time until you reach or approach a plateau. After 7 doses in this example, you're pretty close to that total body stores of 0.75 milligrams that was established by giving a loading dose. So drug accumulation will take place exponentially when you have a constant dosing rate for maintenance and you have first order kinetics of elimination for the drug. Now, there is another approach, of course, to estimate the extent of drug accumulation using this accumulation factor that is shown here. This parameter tau is the dosing interval. I mean, in the case of our example, it was 24 hours or 1 day. And then, of course, you need to know or have an estimation of the elimination rate constant, the first order elimination rate constant for that drug. Now, you can find the derivation of this and other equations in your textbook. And once again, the elimination rate constant that we showed as in the equation for the accumulation factor, estimated as the natural logarithm of 2 divided by the elimination half-life. Now, let's see graphically what happens in 3 different situations here. The first one is that no digitalizing dose, no loading dose was administered, and the drug is accumulating exponentially until it reaches a plateau. The solid line here would be a situation where a loading dose was administered to establish a therapeutic level quickly, and then the optimal maintenance dose was administered over a period of time. Actually, the maintenance dose here is the same as the maintenance dose here. Now let's say that you gave a higher loading dose, twice the loading dose you gave before, but then administer the same maintenance dose that was used here and here. Over a period of time, the concentration that will be achieved at the plateau or when steady state is achieved is the same. So this illustrates the fact that the loading dose does not determine what the concentration is going to be at steady state. And now we're illustrating another useful estimation, namely that 90% of the steady state level with continuous drug administration will be achieved in approximately 3.3 half-lives for that particular drug. Now, practically, think about an individual with normal renal function that is receiving 1/4 milligram of digoxin for maintenance and approaches the plateau concentration in approximately 7 days, as we saw in our example. Now think of an individual with uremia, impaired renal function, and consequently impaired elimination of digoxin. The drug will accumulate, again using the same maintenance dose, and you will anticipate that the plateau concentration is going to be double if the clearance of elimination, say, is reduced by 50%. But the other thing that is important is to recognize that you will not reach the plateau in the patient with impaired renal function until later. This is normal renal function, normal half-life for digoxin. This is impaired renal function and a prolonged half-life for digoxin. Consequently, you will not achieve that steady state concentration until later, in this case, in this example, until 14 days of dosing have taken place. So now let's discuss clearance as a primary parameter in pharmacokinetics. And of course, we need to understand clearance in the context of drug evaluation and use in clinical medicine. Now this is the traditional creatinine clearance equation that you learn in your physiology courses that describes the clearance of creatinine. This is an endogenous product that can be measured in plasma, and the clearance of creatinine being used as an index of renal function. And we have this relationship here that says that U times V over P determines what the creatinine clearance is in that context. So U refers to the urine concentration of the drug, or rather of creatinine in this case. V is the urine volume produced over a period of time. Typically, the creatinine clearance requires a 24-hour urine collection. So this is really a urine formation rate. And then P standing for plasma concentration of creatinine. Now, let's look at this again and think about the appearance of creatinine in the urine, the rate of appearance of creatinine in the urine, dE, think about excretion of creatinine, dE over dt. And now this is equal to the clearance for creatinine and the plasma concentration at that time. So again, that equation that we had before is really a differential equation in disguise. Now let's think about the rate of change of creatinine in the body, X being the amount of creatinine in the body. So we have dx over dt now being equal to I being the rate of creatinine synthesis, this is an endogenous product, minus the clearance of creatinine times the plasma concentration. This would be the creatinine excretion rate. At steady state, we can, of course, discard this term, dx over dt, such that the plasma concentration now is equal to the rate of creatinine synthesis, or is directly proportional to the rate of creatinine synthesis and inversely proportional to the rate of creatinine clearance. And let's look at these steady state equations, because these are truly some of the most useful equations you're going to use in pharmacokinetics. So if we look at continuous synthesis of creatinine, the steady state plasma concentration of creatinine equals to endogenous rate of production of creatinine over the clearance. And if you think about a drug that is being given continuously, say by intravenous infusion, the steady state concentration is going to be equal to the infusion rate over the elimination clearance for that drug. So again, one of the most useful equations for you to keep in mind in addressing what are the determinants of the steady state concentration of the drug. Now, we don't often do creatinine clearance determinations and collect urine for 24 hours, and a number of equations have been developed over the years to estimate the clearance of creatinine in the case of the Cockcroft and Gold equation that has been in use since the 1970s. And you have these parameters here that consider age, that consider weight of the individual, and of course the serum creatinine concentration in milligrams per deciliter. Now this estimate based on the Cockcroft and Gold equation has to be reduced by 15% for women because generally they have a smaller body mass, specifically skeletal muscle mass, and that leads to a reduced estimate for women when using this approach. Now in this equation, or rather in this slide, what you see is that the terms that are shown in red are actually estimating the creatinine synthesis rate that we had in our basic equation previously. An example of the importance of relying on the estimated clearance of creatinine as opposed to simply measuring a serum concentration of creatinine is illustrated in this work by Pierges and colleagues in the early 90s. They had a group of individuals that were clinically toxic due to the use of digoxin. And what they were trying to see is what was the clearance of creatinine in these patients as opposed to the serum concentration of creatinine. And they grouped their patients into individuals that had creatinines in serum of 1.7 milligrams per deciliter or less, or individuals that had greater than 1.7 milligrams per deciliter of creatinine. And these are their estimated clearances of creatinine using the Cockcroft and Gold equation. What you see here is that in the group of individuals with low serum creatinine concentration, relatively low serum creatinine concentration, 19 individuals out of 23 actually had an estimated clearance of creatinine that was less than 50. On the other hand, the majority of individuals with creatinine in serum greater than 1.7 had a clearance of creatinine less than 50. So once again, it is important to estimate the clearance of creatinine. Now, another approach to estimating renal function is based on this equation, the MDRD equation, many versions, that actually estimate the glomerular filtration rate, not the creatinine clearance, but the glomerular filtration rate normalized to body surface area. Now, you're going to have more discussions of this equation in a lecture addressing pharmacokinetics alterations in patients with renal disease. A more modern equation is the CKD epi collaboration equation that is more accurate than the MDRD equation in estimating the glomerular filtration rate, and actually has less bias if the GFR is greater than 60 milliliters per minute, once again normalized to body surface area. So back to our steady state equations. If you have a continuous drug infusion, the steady state concentration is a function of the infusion rate and the clearance of elimination for the drug. If you're using intermittent dosing, say giving the drug once a day or twice a day or whatever the case may be, this is the estimated mean serum concentration over that dosing interval, now being equal to the dose over the dosing interval, and again over the clearance of elimination for the drug. So the steady state concentration, let us emphasize, it is not determined by the loading dose. Now, once again, some drugs require the administration of a loading dose to establish a therapeutic concentration rapidly. But the loading dose does not determine what the steady state concentration will be with continuous administration of the drug. Now, the mean steady state concentration with intermittent drug administration is not determined by the volume of distribution. But on the other hand, we need to pay attention to peak and trough levels because they will be affected by the apparent volume of distribution. And this is shown in this example where the volume of distribution is either large or small, and the same dose being administered over a dosing interval, and you see the variations in peaks and trough, but the mean estimated concentration over the dosing interval is the same and corresponds, of course, to that dosing rate and the elimination clearance. And an important element to highlight is that changes in maintenance dose for most drugs, when we're dealing with first-order kinetics of elimination, result in directly proportional changes in the steady-state concentration, once again, for most drugs that follow first-order kinetics of elimination. We are re-emphasizing our steady-state equations because truly these are equations you should remember because of their conceptual and practical use. But some drugs are not eliminated by first-order kinetics, and I'm giving you 3 examples here: phenytoin, ethyl alcohol, and aspirin. acetyl salicylic acid. These are drugs that deviate from the general pattern of first order kinetics of elimination. And let's focus on phenytoin. Phenytoin undergoes metabolism in the liver via this main pathway of cytochrome P452C9, and we have this para-hydroxylated metabolite that is generated through this pathway. And here we have an example actually from Dr. Arthur J. Atkinson Jr., who had the opportunity to study a patient with phenytoin toxicity. Very high levels of phenytoin observed in this patient upon admission, and with signs of toxicity. Just to give you a reference, the therapeutic range for phenytoin is typically 10 to 20 micrograms per ml. We're near 60 micrograms per ml when this patient was admitted with signs of toxicity. And what they did in this very elegant study is they followed the plasma concentrations of the drug over time, and at the same time they started collecting urine to measure the appearance of this para-hydroxylated metabolite of phenytoin. And you see here day after day after day that the amount of this metabolite of phenytoin that was recovered in the urine remained relatively constant. The plasma concentrations are falling, as you see here, over time, but for a period of time the amount of the metabolite that appears in the urine is constant. And then we reach a point when the plasma concentrations begin to decline more rapidly, and also we see that the amount of metabolite recovered in the urine also diminishes. What this is indicating, and of course they're measuring urine creatinine to validate their urine collections, if you will, over time. And over here they started re-administering phenytoin once again, and they see the increase in the level and then the subsequent decline of the level. What this indicates is that the metabolic pathway, CYP2C9, that generates this hydroxylated metabolite of phenytoin is saturated over a significant period of time because of these very high concentrations of phenytoin that metabolic pathway cannot handle, if you will. Phenytoin kinetics actually follow the pattern of Michaelis-Menten kinetics. Again, concentration over time, in this case given the drug intravenously. This is the rate of change of phenytoin plasma concentration, which does not follow first order kinetics, represented here, or rather determined by the Vmax, that is the maximum capacity of the metabolic pathway, the Michaelis constant, and here again the phenytoin concentration terms. So this is a deviation from first order kinetics of elimination. So let's look again at our steady state equations. We're given the drug, a drug if you will, at intervals orally, and this is the equation we described before for drugs that follow first order kinetics. Clearance of elimination times the mean steady state concentration. In the case of drugs that follow Michaelis-Menten kinetics, like phenytoin and ethyl alcohol and aspirin, you need to apply this equation and this term, if you will, in lieu of this clearance of elimination term. A very important issue, because when you follow this type of kinetics, you lose the element of dose proportionality. Here, the patient receiving 300 milligrams per day of phenytoin has a concentration of 10 micrograms per ml in plasma. Again, the mean steady state concentration. We go up to 400 milligrams and the concentration already doubles. We go to 500 milligrams per day, we have triple the concentration of phenytoin. So we do not have dose proportionality with drugs that follow Michaelis-Menten kinetics of elimination. And again, this is another example of a patient that became toxic on a phenytoin dose of 300 milligrams per day, a typical dose, if you will, but excessive in the case of this individual with slower rate of metabolism. That defining the therapeutic dose then for this patient should really be 200 milligrams per day. And one thing, of course, that arises as a question is, well, there is a large number of drugs that are metabolized in the liver. So an enzymatic pathway is involved, and yet we do not see Michaelis-Menten kinetics for those drugs. So we have apparent first-order kinetics of elimination, and what we can see here is that in situations where the Km, the Michaelis-Menten constant for that particular drug and that enzyme, is much greater than the plasma concentrations that we will need or observe in a therapeutic context in the clinic. If the Km is much greater than C, then we can neglect this term here in the denominator, such that now we have Vmax over Km becoming a pseudo first order rate constant of elimination. So the ratio of two constants, of course, is a constant. So now this becomes the equivalent term, if you will, under conditions in which the Km for that drug and that enzymatic pathway is much greater than the concentrations that we need to obtain for a therapeutic response. So I will refer you to the practice problems that are provided at the end of chapter 2 in our textbook with answers provided in appendix 2 so that you can practice and become comfortable with these concepts. All the equations that I have shown are derived again in the relevant chapters in the textbook. And that will conclude our discussion today. After doing your practice problems and reviewing the lecture material, if you have questions, please contact our course coordinator, who will in turn implement a consultation mechanism with the lecturers in the course so that you can get an answer that will be posted. I hope I have provided you with an overview of our discipline, a critical discipline in the context of drug development and in the context of therapeutic drug utilization. Thank you very much.